What we're doing with those note cards is if you would write on the front the name that you would prefer to be called. So give me your full name so I know who you are for sure, and then tell me if you have a nickname. Next, I want you to tell me what bulimic learning means to you. And if you would prefer to draw me a picture, I always love the pictures because that can evoke some pretty great imagery. Also on the back, if you could tell me one thing about which you're very passionate. I will relate it to micro. So please do be careful because I promise that no matter how outlandish it is, I will somehow bring it in. So if it is sex, drugs, and rock and roll, I will absolutely find a way to relate that. And in fact, the drugs part is soups easy because we will be talking a lot about fermentation, alcoholic and otherwise. We will also be talking about mycology a little bit. So there's lots of room there to talk about those sorts of things. So please do give me an honest answer. I love to know all of those things that get you jazzed, that cream your Twinkie, right? Tell me what they are, and I promise I'm going to bring them in. So I'll be quiet for a couple minutes here. I want you to think about this. And then by the end of the hour, I guarantee I will be all loopy, and I will not remember to ask you to bring your note cards up here and give them to me. So if you all would please remember to just come up and throw your note card on the stage. I have my studying cut out for me, so give me my assignment to learn who you are. And if you want to add to that note card, if there's just something that maybe you just randomly want me to know, I would love that as well. I have no doubt that over the next few minutes we're going to have more people that are swishing in, and that's totally fine. If you feel free, if somebody sits next to you and they're just a little bit disoriented, tell them what to do with their note card and tell them what to do with the syllabus, you know, kind of get them up to speed. I won't mind the annoying whispers, and actually today is a fantastic time to start to tell you that I expect you to be obnoxious in lecture. I expect you to have your cell phones out. I expect rowdiness. I expect engagement. So anything that you have today that you just want to kick that off with, please feel free. I am Rachel, and I'm going to be, it's kind of a dubious honor, I'm going to be your instructor for both lecture and lab, which means that you get to see me 5 days a week. I know. And on Tuesday and Thursday, you get to see me for 2 hours a day. Now, if you think about the fact that that means that all of you have lab on Tuesdays and Thursdays, that means that I'm in the lab from roughly 7 a.m. to 5:30 p.m. So if you ever need to find me on a Tuesday or a Thursday, that is where you're going to find me. On the first page of your syllabus, go ahead and draw your attention down to that set of boxes, and that is all information about me. I like to be called Rachel. I always know if people were here on the first day of the class because they don't call me Dr. Watson, they call me Rachel. And I love that. And in fact, to me, you're showing me the most respect by just calling me Rachel. It's really who I am. But if it just doesn't go for you, you're like, dang, I need a title because it's just a thing for me. I need a title. If that is the case, you can call me Coach. I will always answer to Coach. I have been the coach for the cross-country ski team for 16 years now. It is one of my greatest passions, and it is something that you will hear more about, I promise. We actually were invited to represent the United States this year again in Slovakia at the World University Games. So we get pretty jazzed on that stuff. Now, if you really can't go for the Coach or Rachel thing, I'm trying to get used to Professor Watson. I'm starting to answer that better, little slower, but I will do my best. Now, if you need to find me, two things. One is, again, you will see me 5 days a week, right? So you can ask me questions when you do see me. But I try to be as accessible as possible. On Mondays, I will have office hours twice. The first time I'll have office hours is from 11 to noon in my office. The second time I'll have office hours is online. So you can log in to WYO Courses, hit the conferences button over on the left tab, and you'll find me there online from the comfort of wherever you are, bathtub maybe. Sometimes I log in from the bathtub, and whatever it is that makes you the most comfortable to be in office hours at night with me. So 7:30 to 8:30 p.m. is when you'll find me online on Monday night. Again, remember now, Tuesday and Thursday, I'm in the lab all day. So if you just have a Rachel hankering and you need to come and find me, that is where you'll find me all day. Now, on Wednesdays, I don't have office hours, but on Fridays, you can find me again in the lab from 12:10 to 2. That is, if you're like, man, it's so long to wait for lecture until 2 p.m., I'm going to go into the lab from noon to 2 prior to lecture, and that's a time that we, dork out with a little more micro. So find me there as well. Those are the ways that you can reach me. If you want to email me, that is fantastic. I try to respond very rapidly to email. I do my best to keep up with my email. I will respond to text message, but please do your text message the same way you would an email. That is, tell me who you are. Because if I get some random text from somebody, it just says, did you get that thingy I put in your office? It's like, woo, weird, okay, delete. I'm not going to respond if I get a real weird text with no name on it. So if you do text me, just bear that in mind. Okay, great. There are a few people still coming in. They're like a little disoriented. Where's that note card? Throwing stuff around. On those note cards, please, on the front, I would like your full name, but also your nickname if you have one, how you define bulimic learning, and something that you're very passionate about. Please, disclaimer, I will bring it up in lecture, or I will bring it up in homework, or what have you. So I just want to see, is anybody out there willing to share, as you came to class today, what your primary goal was? Did you come into micro and say, I'm going to, what, goal? Pass? I think somebody said, right? Yes, I am so going to pass, right? That is a great goal, okay? It's an awesome goal. Anybody else come in with that goal in their mind? Okay? Maybe the goal that you have is, I am so going to come out of this with an A. Okay, maybe that was your goal. And it's fine. It's a good goal. Those are good goals. And our system really caters to those kinds of goals, right? We're very oriented to grades. But what I am hoping is that despite maybe having walked in with some of those goals, I hope that you leave with the number one goal of wanting to have a sexy mind. Yeah. Because okay, you guys have had a date like that, right? Yeah, where you're thinking, man, you're real hot, till you open your mouth. Not a whole lot of stairs, right? And there is honest, right? Honest to goodness, there is nothing sexier than being passionate and knowledgeable about the subject matter that you choose to study. And that's what I want to convince you of. We have a lot of derogatory terms for people who like to dork out with nerdy material. Like we like to call them nerds, right? And, that's fine, but honestly, those are the sexiest folks around. I mean, when you can talk in an informed way about, oh, come on, face it. When you can talk about how your microbiome dictates your health, not only your physical health, whether or not you're prone to obesity, whether or not you have high cholesterol levels, and also, get this, you guys, there is a brain-gut axis, which actually means that your microbiome dictates your psychological health. And that is cutting-edge stuff. If you can dork out with that, hot, right? Okay, so I want you to hopefully leave my class with that becoming something that is your primary goal, much ahead of the grades that you receive, okay? So that being said, what are some tools in micro that can allow you to get a sexy mind? Turning to the top of page 2 in your syllabus, you're going to see that there are a few things that you might want to purchase for the class. Number 1, the one thing that you absolutely have to have is this. This is the micro lab manual. So, unfortunately, even if, you're at the grocery store and you're trying to decide whether you can afford beer or toilet paper, and you're like, oh man, but I have to purchase the micro manual. I'm sorry, you're going to have to put back the beer, hopefully get the toilet paper, and you're going to have to buy this. Okay, this is absolutely required. Who's already gotten it? Nice. You guys so rock my world. Okay, so this is absolutely required. Now, everything else for the class is not necessarily absolutely required. However, it is suggested, heavily suggested, that you get the textbook, and we're doing the online thing, and this is kind of fun, means a little less paper waste. I like the green option. If you're just old-fashioned and you like the smell of the book and you like to just stick your nose in it and that's about all you do with it, but no. But if you just want the old-fashioned variety, that's fine too. But I've decided this is the semester of options. I'm really into options this semester, and so here's the option that I'm going to put forward to you. If you really like the Learn Smart courseware that goes with the book, you can purchase that package, and if you do it throughout the semester, there are just these little exercises that go with each chapter, and you submit those. I will allow you to use that to replace your low exam score. So if you just decide that's a great option for you, maybe exams are not your cup of tea, you might want to do this. So it's something to think about to purchase this from the bookstore. Find your way to this site, which I've written up here, and this is also on Wild Courses so that if you don't want to jot it down now, you don't have to. And begin to do those exercises, because you might be thinking, hey, I bet I might have a day where I don't really feel like taking an exam, put in a 0 and replace it with this. Or you're a little worried that you might have a lower exam score. Okay, now I'm going to come with another one of those options here in just a moment. So if you purchase the textbook, that might be a great thing. The other thing that I'm going to put out there is this summer I was bike touring. I rode from Banff to Jasper over the Icefields Parkway, absolutely rocking. If anybody has a chance to do that, you should absolutely do that. But I started really personally feeling affected by climate change because I got to see the rings of glacial recession. I started thinking about the way in which that related to microbiology, the ways in which infectious disease spread are changed by climate change. And so I think right now it's pretty poignant because what's going on in Africa? Ebola. Might that be related? You bet. So what I decided is, wow, I wonder if there's a sexy-minded book out there that talks about the interfaces between climate change and the microbial world. And I found this book called Fevered by Linda Marsa. And you can purchase it for your Kindle. You can purchase it on your iPad, read it on your phone or something. And I highly suggest it. It is a fantastic read. It is absolutely poignant with regards to what's going on right now with Ebola, but also many other impacts potentially of climate change. So if you grab this book and you start reading it and you get jazzed on the discussion that we're going to have online, again, you can replace your low exam score. So this is another option for getting rid of the exam score that you don't like. I also just say, sexy-minded read, get it. Because, it's just going to be fun to discuss it with all of you and to see the kind of diverse, discussion that comes up around it, okay? This is, again, on your syllabus, so you can definitely find it on your own time. It's not a bookstore book. It's just something that I would find, honestly, it's real cheap on Amazon. I didn't say that. So who got my email about the website? You found your way to the website? Okay, good. So I want to just show you like a little bit around. I have a feeling a lot of you have already done this tour on your own. But notice that if you find that you lose the syllabus immediately after leaving class, understandable, it is on here and you can find everything, including the schedule on here, so that you know what is up and coming. One thing that I don't like is I don't like being bound every day in lecture to this railroad track schedule. I mean, what if one of you asks a really awesome question? And then I'm like, sorry, no time for that. We got to railroad along, I don't like that very much. So what I don't hold us to is the content schedule that I promise will cover everything in one way or another. But what I do hold us to is the scheduling of exams and homeworks and those sorts of things. So this is something you want to go hang up somewhere so that you do know when those dates are going to take place. And that's very important, and we'll stick to that. Now, also on the web page are what are skeleton outlines for the lectures that we will cover. These are PDF format, and they just provide a little bit of the framework of the notes that we're going to use on that day. And I saw a couple of you already had those, and that's fantastic. So you're thinking, yeah, this is rad. All I have to do is go to Micro and fill in the blank and A. No. Yeah. You got to engage. You got to write outside the lines too. You've got to really get jazzed on what we're talking about, and then we'll be talking about those A's, okay? So definitely don't look on these as just the cheat sheet for getting through the class. They're really just helpful to you so that at times you might be able to have a chance to look at me rather than like scribbling the whole entire time, which is nice for me because, you know, you guys are good looking. All right, so, oh, okay. So let's just talk about the perks, if you will, of coming to lecture. And this is confounded, but I want to share with you, I've just looked at class attendance and exam score and what we see there, because what I should tell you is that you might notice I'm wearing various microphones today. The lecture is being recorded in multiple forms right now. It's going to go onto YOcast. I also will upload it onto YouTube. I will then embed it into the shell. So you'll be able to find absolutely every lecture there. So if you just have a hangover, which by the way, don't have a hangover at 2 p.m., you guys. But if you do, right, if you do and you miss lecture, you can watch it later from the comfort of your bathtub again. But that being said, right, okay, so that option, it is out there, right, it is out there. But I think that coming to lecture is your best bet. These data would somewhat support that. Those students who were there, both the times I took attendance, had a 76 average on the exam. Those that were only there for one of those had a 69. Those that never did attend had a 63.7. So what we might say is there's a correlation between class attendance and exam score. Those of you who are really dorky about statistics, maybe you had Stat 2050 this morning, you're like, Rachel, correlation doesn't indicate causation, right? So it's hard for us to know. Maybe, in fact, the people that came to class were just smarter anyway, and so they would have gotten a higher score anyhow. Yeah, that's true, but it doesn't hurt to kind of engage and come to class, right? So that is just one note. Homework is going to be graded this semester. I will give you points for doing weekly homework assignments. Already at 5 p.m. today, your first homework assignment will open on YO courses. So let me show you around just a little bit on YO courses. The primary button that you will need to use is the modules button. When you click on that, it's going to bring up the homework assignment, and as I said, tonight at 5 p.m., homework 1 will open. You can then click on homework 1, and from there you'll find a PDF file that you can download, and then you can do your homework when you come to class. For example, you can download it, bring it with you in any form you like, and you can complete it while I'm lecturing. In fact, I even encourage you to be completing it while I'm lecturing and to stop me at a moment's notice and say, Rachel, seriously, you did a really bad job of covering question number 4. Could you please go back and help with question number 4? I'm more than happy to do that. I like it when I see that you're kind of working on the homework simultaneously with the material that we're covering. So that enables you to finish your homework. You could come to my office hours and get help. You could do it in lecture, and you can absolutely ensure that before you submit the homework, you have 100%. Because then when you're ready to submit it, you'll just click on the Take the Quiz button, and all it is an answer sheet, and you'll just fill out the answers that you've already got from having finished the homework earlier. Does that make sense? Okay, cool. So that's how that's going to work every week. Tuesday is a big day. So if you have your phones with you right now, just go ahead and put a standing, alarm to go off every Tuesday that says micro something today, There will either be a homework due on Tuesday or you will have an exam on Tuesday. So just make note that those are the days that you're going to want to think about micro. Okay? So homework's a big part of what we'll be looking to assess you on this semester, but not the only part. And that is to say that there are exams, traditional exams, but like I said, there are options for replacing those. Also, we have the most rad assignment of anywhere, I think. It's the Poster Current Events Project. And just as you all are watching this Ebola outbreak, we're seeing how trendy microbiology is. In fact, I would argue that it is the most trendy science right now. So if you get into the news and you start watching, you're going to see so many things that relate to micro. So tomorrow in lab, we are going to form groups, and you're going to get to know your group. You're going to be like, you have a sexy mind, let's get together. And you're going to begin work on a current events poster assignment. This is an assignment that goes throughout the entire semester. Don't be deceived. Although it is presented during our regularly scheduled final exam, it is done well before that. That allows us to make the regularly scheduled final exam time a party, where basically you present your poster, we're pretty chill, we all come around and say hi, and we talk about it in a somewhat informal setting. Your posters will be done well before that. Has anyone been so sexy-minded to have already watched the vodcast on the poster? Nice, nice. So if you have not, that is your #1 goal for tonight. Go ahead and download that lecture. Actually, it's right on YouTube, so you just click on it right there in the shell. You're going to find that. And in fact, I can show you. I'm going to show you where this is. Because here's what I'm thinking. What do you think? I think we should have like a very informal little quiz on Friday in lecture that will be about the poster vodcast. So right here it is, current events poster assignment description. You click on that and you're going to see 2 YouTube videos. They're just split into 2 parts for your viewing pleasure. But in any case, if you watch those, I guarantee you, some extra credit on Friday for just taking this, little quiz of extra credit. I'd really like everyone to know what's going on with the poster assignment, okay? So that's your #1 objective for right now. So that is going to be another place that you derive your points. You're also going to get points from lab. Lab begins tomorrow, and starting tomorrow, I will see you every Tuesday and Thursday, excepting the Tuesday prior to Thanksgiving. We're going to take that off, and we'll end a little early with labs. Any questions about the syllabus at this point? Because I have jetted through it, and I have already taken us to the bottom of page #2. So some of you have probably done some really creative drawings by now for your definition of bulimic learning. Anybody willing to share a time that you learned deeply? I would consider deep learning to be the opposite of bulimic learning. What leads to deep learning for you in your life? You guys are going to get way more bold by the end of the semester. Yeah?
I'd say repetition.
Yeah, great. So repetition is definitely a thing that helps us to retain information. That is to take things from our desktop to our hard drive, and so to speak. So repetition can be a good way to improve your learning. Anybody have something else? Yeah. Is it Lauren?
Lauren.
Nice.
Teaching someone else about the subject I'm learning about?
Yeah, totally, right? So you just hit on it big time. Now, I don't know if many of you are Harry Potter fans, but I kind of get jazzed about it. And, I actually backpacked down into Volcanoes National Park carrying the hardback version of the Harry Potter book that talked about the DA Club and the Room of Requirement, right? And I mean, this is such a thing because really what that shows is that when we teach each other, right, when we actually engage in teaching someone else, we learn a lot better. And that's a big part of the poster group that we're going to assemble. I think always thinking about that is great because, you're going to get somebody in your poster group, right, that you think, man, you waste of space, right? But think about Neville. Who saved the day in the end? You, right? Seriously. So I think it's always good to think about everyone in your group has something to contribute. And if we start from that point of reference today, I don't think we'll ever go wrong. Okay. All right, so current events poster, assignment description, exams. Last thing I'm going to talk about is page 3 on the syllabus. And exams are dictated there, but I want to let you know that although in the formal scheduling book this is down, what I like to do is to give you a window of opportunity for exams. So they are traditional exams. They're closed book, closed note, kind of. The third one has a poster that's for metabolism. It's open reference, kind of. But you're going to come to the lab anytime between 7:30 a.m. and 7:30 p.m. preferred. And you're going to take the exam at your leisure. So if you want to come in at 7:30 a.m., you want to sit there for 4 hours, you want to take a bathroom break, come back for another 3 hours, you can do that. I don't advise it, but it's allowed, okay? So there's just a lot of flexibility with exam taking, very different in that way. So anytime on those days, and you can note down exam 1 through 3 and the dates as they're already scheduled. Now, you will have taken note that I mentioned that our regularly scheduled final exam will be a time that we dedicate to the poster presentation. So that means that we must not have a traditional final exam. We do not. We have a take-home, open note, open book, open resource of any kind final exam. But don't be deceived. It is the most sexy-minded assessment you will ever undergo. And the good thing about it, right, is that the whole time you're going through and you're like, oh man, I just rocked that. Dang, it feels good, you know, to see how your intellect has grown. But it will definitely be challenging. It will definitely be challenging. Makeup exams, I'm hoping with these options out there that we won't have so many makeup exams. But if you do need to have a makeup exam, we'll deal with it at the time. Please, communication is key for me. I really, really want to know why something's going on that's preventing you from being in class. So if you could like write or an email or call me or text one of the TAs or send smoke signals, I don't care. As long as you're communicating with me, I promise I will work with you. I mean, if the excuse is as ridiculous as I just have to take a vacation for mental health to Mexico, you know, I'll be like, okay, you know, all right, you told me ahead of time, let's work with that. Let's figure out how we're going to work with that. On the other hand, if you come back with a suntan and you're like, so tell us how am I going to get caught up now, Rachel? It's like, not so much, because you didn't even bother to send me smoke signals. And then I'll be, you know, I'll be jealous of your trip at that point and things won't be good. Okay, so grades, there's one big change on grades this semester. We're going to go to the plus-minus system because eventually we're going to have to, and I've decided that we should just go ahead and do it, right? Might as well just be early adopters on this one and just get it done. So you'll notice that that is looking a little bit different than maybe it has in the past. I have a couple of pet peeves, and I will be so impressed if you guys can remember these come the end of the semester. Number 1. Please act early. If you know you need to have a B in here in order to retain scholarships or in order to get into med school or whatever it is that you need to do, make sure that you communicate that to me right away and that you show me that you're willing to do what it takes to get that grade even before the first exam. What I can't stand is when it comes time to submit the final, it's the day before, and you come in and you're like, Rachel, I have to have a B. I know I have an F right now, but I have to have a B. And then I'm like, I can't help you. Like, I can bend over backwards, but there is nothing I can do right now. However, if you had come to me after the first exam, there's a lot I could do. And you don't tie my hands. Other pet peeve is when the final grade is submitted, everything is done, right? I put in that final grade, and you have an 87%. And you email me and you say, Rachel, is there any way that I can get those last 3 points? Any math majors? Yeah, 87%. It's like, this is a class that is worth 800 points. It's not 3 points that you need there. Right? So please show me that you're at least doing the math so so that you know that that was like the difference between getting a 60 on an exam and an 85 that would have actually given you the next grade that you needed. So it's not 3 points. So show me that you at least know how you're doing the math there when you email the argue points with me at the end of the semester. So when do labs start? Tomorrow. Yeah, I'll see you tomorrow for labs. Now I think we're ready to get started on the fun stuff. Every day before I begin lecture, I'm going to make announcements, and I'm going to try to always remind you of the up and coming events. So if you happen to come in late, totally fine, but make sure you go back to the vodcast and review what that day's announcements were and that you're on the up and up with all of that. One of the things that you're going to see in the YO courses shell is the option to take a pre-test and knowledge survey. That pre-test and knowledge survey is to allow us to see how our students as microbiology majors are progressing through their degree and learning the material that we would like them to know when they graduate with a microbiology degree. Which means that as you guys go through this pre-test, you're not going to know most of it. And that's okay. I just want you to give me your honest feedback on it and to let me know that you don't know it, and then to accordingly rate your confidence. So if you don't know the answer, tell me you're not confident, right? So that's what I would like you to do if, it's optional, completely optional, if you would like to know that at the end of this semester I will drop your low homework score. That is, I'll just replace it with a 5, a full 100% on that homework score. So if you get in there and you go ahead and take that pre-test and knowledge survey, that is how we'll give you a little bit of incentive for that. But the other kind of cool thing about it is that you'll see some questions that at the beginning of the semester you'll be clueless on, and then by the end of the semester you'll be like, man, I totally rocked that. And that's kind of neat to see how you progress through the semester and how you know some of the content that maybe we're going to cover. So it's good for everything, not just for our understanding of your learning, but for your own understanding of your own learning. So homework 1, I mentioned, that's going to be due next Tuesday. It will open tonight at 5 p.m. Watch the current events poster project. We talked about that, the YouTube videos that are up, and labs start tomorrow. There is a tutoring center that's opening up just this year. The tutor is fantastic. He's actually been a TA of mine for many years. So take advantage of the STEP Tutoring Center if that's something that works for you. And you just go to the research help desk. Starting, I believe, next week is when they're going to start to have scheduled tutoring over there. So that's so much for the boring announcements. And I have now a more fun question for all of you. That is, when did scientists finally fully support the idea that life comes from other life? I mean, this is probably something that you've known since before you were in kindergarten, right? That babies come from other living organisms, right? That life comes from other life. But it took scientists a very long time to actually disprove the notion that things could just pop up from inorganic material. That is to say, they thought that if you had this sterile broth in here, right, which is a nutrient medium that organisms like to eat, good food, that bacteria could pop up in here spontaneously from nothing. Abiogenesis would be another term for that. So what I want to know here is when did we finally put the nail in the coffin of the term spontaneous generation? Who's got a thought? Yeah. Thinking 457 years ago. What's your name? Esteban. Nice to meet you. So Esteban's got a guess. He's like, well, you know, it seems like, gosh, that's pretty, you know, it's got to be a while ago that we realized that life comes from other life for sure. So 457 years ago. Anybody want to give an arguing point to Esteban? Yeah? Okay, maybe 138 years ago. Maybe it honestly wasn't all that long ago that we realized this, right? It seems like, wow, it blows the mind. What's your name? Amy, nice to meet you. Awesome. Okay, so yeah, what about Louis Pasteur? You know, she's got some great ideas. So let's go dive into the Amy Esteban debate. And let's find out whether or not this was 457 or 138 years ago. And let's begin 4.6 to 4.5 billion years ago. That's about how old the Earth is. That is the estimated age of the Earth. And many people have debated how it was that the first cellular life form came about on this ancient Earth. And right now, one of the current hypotheses that is put out there is something called the RNA world hypothesis. This says literally that RNA was the first molecule to both act as the code, the genetic code, the primary genetic code, and the catalyst. That is, it could have dual roles as both an enzyme and a nucleic acid. And this idea has gained much speed, although there are some contentious ideas that maybe it wasn't RNA doing everything. Maybe metals acted as catalysts and RNA did some of it, right? But this is a very famous hypothesis, the RNA world hypothesis. Now, about 1977, in Australia, there was a fossilized organism very similar to a cyanobacterium called an archaean chert that was uncovered. This was found to be 3.5 million years old, and this was the first kind of record of life. So microorganisms are thought to have been around for 3.5 billion years, but honestly, we haven't known about them for very long at all. And in fact, it wasn't until Antony van Leeuwenhoek, who was actually a draper and a haberdasher. Anybody know what a haberdasher is? What that is? It's a maker of men's clothing. So okay, this guy's literally a superhero. By day, maker of men's clothing. By night, microscopy. So he had a sexy mind. And what he did was he designed lenses. And he was able to make a microscope lens that had a short enough focal length that for the first time he was able to focus on the microbial world. And he was jazzed. I mean, he was real jazzed. And you know what? He started taking samples from everywhere he could find. He would run around and take pond water. He would go to the restroom and sample urinalysis. He would just check anything he could and look for microbial life in it. And so this was around 1673 that he began to write about his findings and seeing the microbial world through a microscope that could magnify some 50 to 300 times. Now, this was more powerful than the lenses of either Galileo or Hooke. It was unprecedented. And he began to write letters to the Royal Society. And I'm going to read to you from one of those letters as you ponder what it must have been like to see these tiny animalcules for the first time. And that's what he called them, little animals or animalcules. And so he wrote to the Royal Society, and he said to them, very many little animalcules, whereof some were roundish, while others a bit bigger consisted of an oval. On these last I saw 2 little legs near the head and 2 little fins at the hindmost end of the body. Others were somewhat longer than an oval, and these were very slow moving, few in number. These animalcules had diverse colors, some being whitish and transparent, others being green with very glittering little scales, others again were green in the middle and before and behind white, others yet were ashid gray. And the motion of most of these animalcules in the water was so swift and so various, upwards, downwards, and round about, that it was wonderful to see. Jazzed, right? He was so jazzed. And you know, it's amazing, even now, when we look at a drop of pond water and we see these sorts of things, I guarantee you guys are going to be pretty stoked when you do this next week in lab. And we know they're there. So think about not knowing that there's this whole other world and observing it for the first time. So Antony van Leeuwenhoek is sometimes called, for that very reason, the father of microbiology. And we might define microbiology as the study of life forms and agents that are too small to be seen with the unaided eye. Now, like every single definition out there, there are always things that can't quite fit into the definition. I mentioned that we would talk this semester about fungi, and we know that many of those are not microscopic, but yet we study them as a part of microbiology. So not everything is too small to be seen with the unaided eye, and really crazy enough, there is this amazing prokaryote, a bacterium, that is found off of the coast of Namibia. It was discovered in 1997. It's called Thiomargarita namibiensis, and it's a bacterium that can be seen with the unaided eye. It's so big. That's how big it is. So that's just crazy, right? And so obviously it sits outside that definition as well. Notice that something interesting here, if you had taken the class when I first started teaching microbiology, I wouldn't have put that word agents in there. Does anybody know to what that refers? It's not a life form, but it's an agent. Yeah, viruses, what else? Viruses are a big one, right? And so when we think about Ebola, Ebola is not actually a life form, it's an agent, meaning that we don't see it having both DNA and RNA. So it is categorized as an agent rather than a life form because of that. Now, still, we could debate it, right, whether viruses should be put in that category, but they are. The other things that are in that category are prions, for example. These are proteinaceous infectious agents. You guys have heard of CWD, yeah? Chronic wasting disease. Maybe you've heard of BSE or mad cow disease, right? These are proteinaceous infectious agents. They're agents. They're not life forms. So microbiology can really be broken down into the study of both those life forms and agents. And this picture is kind of dedicated to reviewing that. And so I just want to make sure that we kind of jot down what are these life forms versus what are these agents. So for example, bacteria, and what you're seeing on this picture here is a slide that will look very much like one of the slides that you're going to do in lab, where you're seeing bacteria and you're seeing yeast. Yeast are fungi. So the world of bacteria and the world of yeast or fungi, these are life forms. This is actually Saccharomyces cerevisiae that I'm labeling there, the kind of roundish looking ones. And we're going to see those. They're the baker's yeast, which are just, we basically take a packet of Red Star yeast and look at them under the scope. This is a protist. It's a life form as well, so protista. These are life forms. We also have another domain called archaea that's not pictured here that is a life form. We kind of think of them as being the ancient ones. The archaea are deeply branching. We often think of them as being extremophiles. We'll talk more of that domain of life. But of course, we also have the agent. And as you guys talked about viruses, now this isn't a picture of Ebola here, but this is a picture of the influenza virus or the HIV virus. And then this is a picture of a prion. And then this is actually a viroid. Viroids are weird. They're actually RNA, and they're infectious agents of plants. There are also something called satellites, which are RNA or DNA, and they're just nucleic acid. So there's so much out there that fits into this microbial world that maybe kind of pushes the bounds of what you thought might be studied in microbiology. There are a lot of different things. So life forms and agents. So, but hang on, I don't feel much closer to, establishing the answer to the debate right here. So we've got to get the Esteban-Amy debate going on. So that's where we need to go at this point in looking at the history of microbiology. So spontaneous generation, this is that idea that life just pops up from unliving stuff as something we know is not the case. But so many held to this idea for a very long time. And so scientists were pushed to either be on one side or another of this debate, and many wanted to support the notion of biogenesis. Biogenesis meaning that one life form comes from another life form of the same type. And really, the debate should have been laid to rest with a man named Francesco Redi. I get pretty jazzed about good old Francesco because he turns out he's a Florentinian, which means he was born and did his work in Florence. And this last year, I had the great opportunity to take the ski team to the World University Games in Italy, and we traveled to Florence, and I was like, oh my God, that's a statue right there of Francesco Redi. So I had like all the poses, you know, I was setting up and taking a lot of photos of Francesco. But this is him. He had the nice 1600s look, but you can see there that he certainly had a sexy mind. Hopefully that's coming through in the statue. But what he did was that he had an experiment set up where it wasn't very tricky. What he had was a slab of meat. Forgive my beef jerky. That's all I didn't want to bring a slab of meat. So we've got beef jerky in here, but he had a slab of meat in there. He put it into two kind of situations. He had one where it was open to the air, and he found that flies got in, they laid their eggs, and there were maggots. And the meat putrefied, right? However, if he put the lid onto that, clearly what didn't happen? No putrefaction, right? Now, what he also did was he put gauze on the top. So now what did the flies do? They could smell the meat, so they laid their eggs on the gauze. So he was actually able to show that flies beget flies, right? That was the point of that experiment. And so with that experiment, we should have been like, oh, cool, life comes from other life. But that's not the way things went. People still held to the notion of spontaneous generation because what wasn't Francesco Redi able to show? What wasn't he able to prevent the growth of? Bacteria, microorganisms, right? And so because of that, people held to the notion of spontaneous generation for a very long time. So although there were many experiments done that would show that if you had broth in a sealed flask and you boiled that broth, that you wouldn't see any growth. So right now, this has been autoclaved. It's been boiled at a very high temperature, high pressure, and it's not going to grow. But proponents of spontaneous generation said, oh, well, when you put the cap on there and you boil it, that gets rid of the vital force in air that might be there to allow spontaneous generation. And so it was very hard for researchers to disprove the notion that there was a vital force in this oxygen that kept getting in there whenever you would open the cap. Because every time the cap got open, life would form in the broth. So these debates went on for a very long time. They included things like a man named Theodore Schwann would like heat the air in a tube before he let then it be exposed to the air. But then the spontaneous generation proponents said, well, when you do that, you're getting rid of the vital force in the air, so that's not a viable experiment. And so the debate continued for many, many years. And until the work that Amy brought up, which is that of Louis Pasteur. So Amy, I think you've got something on your ideas of what Louis Pasteur did. Go ahead and tell us a little bit about that.
I can't remember what the flask. We had
Yeah.
something in the flask.
Mm-hmm.
And we had a weird straw that came out and it's perfect for like to let the air in so the spontaneous
Yeah. It allowed the air to come in, and so if air contains some vital force, wouldn't it allow the growth? And so what she's saying, he had kind of this funny shaped straw. Let me show you the design of this. It's actually called a swan-necked flask. So the design that he put forth was this one here, where what would happen is in here, and what he had was boiled beef broth, and it was sterile. But in order to show that air didn't cause growth, he allowed this long kind of swan neck to bring in oxygen. So air could come in, and if there was in fact a vital force in that air, it would come and be exposed to the broth and allow for growth if there was this vital force. But in fact, what happened was that dust and microorganisms settled out in the first bend of the flask. And so there was no vital force in the air that allowed for spontaneous generation. So Louis Pasteur had the brilliance of being able to show that exposure to air alone didn't allow for spontaneous generation. However, if he broke the neck of the swan, he could then see growth in that boiled broth. Now, what does this indicate about what is present in air? If dust settled out in that first neck, go ahead. What's your name?
Zach.
Zach, it's nice to meet you. So Zach, I think you've got the answer.
Microorganisms?
Yeah, you got it. Louis Pasteur showed that microorganisms were present in air. And that was what was causing growth, not some elusive vital force that was present there. And so his genius was twofold. One, showing that there were bacteria in the air. Two, showing that the air alone did not have some vital elusive force that allowed bacteria to pop up from nothing. So with that in mind, I want to leave you with one thought today. What now would need to be done to fully close the gap to disprove the notion of spontaneous generation? What would other researchers need to be able to do to allow this idea to just firmly go away? Under the notion of the scientific method, other researchers would need to be able to what? Go ahead. To repeat the work. Okay, so what's your name? Rex, got it. All right, Zach, Rex, Amy, Esteban, Lauren. Okay, I'm getting there, and I will know way more after tonight. You guys are bringing your note cards up to the stage so I can study, and have a fantastic afternoon.